Welcome to the Kellogg's Origins soils tents. My name's Duncan Rawson. I am senior partner at a consultancy business called EFFP. But the reason I'm here today is because one of my clients is Kellogg's. So I'm here to represent Kellogg's and their Origins program today. The first thing I need to say is that we're actually in between sustainability managers at Kellogg's at the moment. And so we don't actually have anybody from Kellogg's. So they send their apologies, but really hope to be with us next year. Kellogg's and the Origins project are really delighted to be sponsoring the soils tent. It's the second year that we've done it. And the reason we do it is it fits very, very closely to the work that we're doing through the Origins project with our farmers, helping them and encourage them on their transition to more regenerative farming practices. And what we're going to do today is spend a bit of time just showcasing some of the work that we've been doing over the last couple of years in particular. The title of our session is Growing Resilience. And with everything going on at the moment, and I don't need to tell you what's going on at the moment around the world, I believe with some passion, and Kellogg's also believe as well, that we need farming systems that are now becoming more resilient, managing risks in ways that they haven't had to manage before. So again, a lot of the work that we're doing is about trying to build resilience into farming systems. I believe it is a very, very important thing to do. So what we've got today, we've got two sort of sessions, and they kind of overlap a little bit. We're going to start off looking at some of the work that we've been doing around nitrogen utilisation over the last couple of years. We have got some booklets at the front if you are more interested in some of the granular data that's coming out of those, so come and pick those up. And we're starting off with Hugh, Martineau, he's there. Oh, we're starting off with Clive, apparently, and then moving on to Hugh. Now, they both spoke last year at these very events about some of the work that we're doing, so there's a good opportunity to just provide an update of where things have got to. From what I can see, it's fantastic work, but it kind of begs more questions than it answers a lot of the time. After that session, that'll be for around about 20 minutes or so. We're then going to move on to Alastair Leake, who'll be known to many of you, I'm sure. Alastair's just involved somewhere else at the moment, but he promises that he'll be back ready for his session. He's the policy director at Game and Wildlife Conservation Trust and also the farms director at Loddington, their demo farm up there as well. And he's going to be talking about two things. He's going to be talking about some of the work that we've been doing on habitat management and how we're encouraging the right pests to be on the farm at the right time, and also talking about his hedgerow carbon project that he's just recently started, and Kellogg's delighted to be able to support that process as well. So that's it from me. Sorry, the format, we'll listen to the two presentations, as it were, and then we'll have a Q&A session at the end where hopefully we'll get some views and questions from you as well. So I will stop waffling on, and I'll hand over to Clive, and then for the first session.
Thanks, Duncan. So I'm going to talk to you a little bit this morning about the work we've been doing with the nitrogen use efficiency. I'm going to talk about the agronomic side, about how we've been applying the nitrogen and making those decisions. And I'm going to hand over to Hugh, who will talk about the carbon aspects of the data we've been monitoring. So nitrogen use efficiency isn't a metric that's typically used on farm. And it's pretty crazy that as an industry, we really don't know how use efficient we are with our most expensive product and input. And typically in the UK, we're probably the leaders in the world in nitrogen use efficiency, and typically we're only 64% nitrogen use efficient. And there's an article I found in Farmers Weekly that was sort of highlighting that about the use efficiency, and it's quite frightening. And when I started measuring our use efficiency on the farm at home with my brother, we were typically in that 64% use efficiency bracket, and it just horrified me to think that of the investment we were making, 36% of it I couldn't account for, and I didn't know where it was. So trying to understand how I could improve that use efficiency, how I could maintain that nitrogen, get it into the crop, rather than lose it into the environment, was really, really the main crux of what we were trying to do. Working with Mapavag, so Mapavag is a data business. We've developed a pure farming platform that allows us to ingest data and pull data together, on the back of which we can then add value with different models. And for the Kellogg's Origins project, we've actually built three different products that have come out of this in terms of, firstly, a benchmarking system to show farmers where you are with nitrogen use efficiency. So if you don't know where you're starting from, you've got to start and measure. So we can then do that very quickly. We can benchmark where you've moved from, then we can benchmark progress, and then evidence the change we've got, whether it's positive or negative. The second aspect of that was then that the decisions we have to make with nitrogen are always a challenge, and it's always a grey area, trying to understand and interpret nitrogen uptake by crops, how crops take nitrogen up and are using them every season varies and changes. So having a system in place that will allow us to augment those decisions and support them is really important. So we've built a system to really support the growers and farmers with technology that allows us the opportunity to use the plant as a sensor and then inform our decision-making process on nitrogen. And then thirdly, obviously, climate change is hugely important. Carbon is a very significant part of that. And being able to benchmark and measure carbon aspects of where nitrogen particularly sits, as it's our biggest greenhouse gas emitter within agriculture, or certainly arable agriculture, it's really important to understand that as well. And I'm not going to talk any more about that. I'm going to leave that to Hugh. So where we started was, we started 2 years ago now. The first thing we did was benchmark a group of the Kellogg's growers. And what we found initially was that there was a huge change in use efficiency. So nitrogen use efficiency on some of the farms was as low as 45%. In other farms, it was 120%. And when I'm talking about nitrogen use efficiency here, the measurement we're actually doing is fertilizer use efficiency. It's the nitrogen we apply to the crop to the nitrogen we then remove with the crop. So if we're at 120% use efficient, what that actually means is that we're actually taking nitrogen out of the soil, from the bank of soil, and taking it away with our crop. And that's something we need to understand and manage and measure in better conditions. So from that base work, we then started monitoring crops. And essentially all we've asked farmers to do is use very simple technology. So technology that's been around for 20 years. I'm sure some of you have used them, some of you have seen them. This is just a Yara N tester. It's a chlorophyll meter, and it basically measures the amount of chlorophyll in the leaf. And the theory of what we're trying to do is using the plant as a sensor. The plant's in the environment where it's taking the nitrogen up. If we can measure how much nitrogen is in the plant and where that nitrogen's going, we can then measure how effectively the plants are using it and when they're becoming deficient. So we can do two things with that. We can firstly make sure that we don't oversupply a crop with nitrogen when it doesn't need it. But secondly, we know when the crops need the nitrogen, so we can apply it in an appropriate timescale and make sure that the crops will utilise what we put on and don't cause extra losses. We supplement that in the first season with leaf tests and with grain samples at harvest, so we had the grain protein and the full nutrient analysis of the grain, so we could benchmark where we were and make sure nothing else was limiting nitrogen uptake. But the learnings we had from the first season, we've sort of supplemented it a bit further. The challenge we found with very high nitrogen use efficiencies is we didn't know what the impact on that would be on soil organic matter, and whether we could measure that, and whether that was going to be a challenge and could be detrimental to the quality of our soils if we're continually applying less nitrogen, but in the process degrading soils through utilising what's in there. So ultimately it could leave us in a worse position. So making sure we understood and maintained that was equally as important. So for this season, we've introduced soil testing on some of the sites. We've also put soil temperature and soil moisture sensors in, because what we found a couple of years ago was that nitrogen uptake was being restricted in cold temperatures, and being able to monitor those temperatures and water availability on site are really important for helping with yield prediction. The theory is then we go into the field, and on a weekly basis we've been asking the farmers to take a SPAD meter reading, and literally we've just been documenting that. And you can see on the graph here the red dots are the leaf nitrogen readings as taken by the N tester. You can see that I've got a lot of deficiency in this particular crop. We were limited on water, so we hadn't made an application. We waited for a rainfall window that supported an application, and you can see with 2 or 3 mils of water that you can actually see that nitrogen getting into the plant within 3 to 6 days. So that was 2 years ago. That was 2020. And it was quite interesting to see how quickly, with so little water, nitrogen could get actually into the plant in the right conditions. What horrified us last year was thinking we were quite smart, that we had quite a dry spring, was when we did the same process last year, waiting for a rainfall event. Because soil temperatures were so cold, it delayed nitrogen uptake. The biological process that's used by the soil and the environment and plants to get nitrogen from the soil into the plant was being impaired by the soil temperatures. The biology couldn't work to full efficiency. And we were seeing that last year particularly, it was taking 22 days, and in fact it was before a soil temperature change of above 10 degrees really occurred before we started getting nitrogen into the plant. And this has two effects. We can measure soil for nitrogen, and we can measure it early in the season. Interpreting what will be released in that growing season is a really dark art. And understanding and predicting that, there are some ways of doing that, but it's still a very dark art and expensive to do. So having a tester that you can use regularly to take those readings really offered us a lot of opportunity. We then benchmarked this with the farmers, and this is just a case of 3 fields on one particular farm. What we did wrong last year was we didn't replicate things in field properly. So this is an example of where we tried it in one field versus 2 other fields that were neighbouring with the same farm practice. And, you know, we were quite happy that the nitrogen use efficiency on the farm stuff was a lot lower than what we were doing. So we could see an improvement in use efficiency for sure. What we've done this year is replicated every field, and every farmer's got a trial tram line or 2 in there that we can then take to yield at harvest, and we'll take protein samples from each. The main benefits and improvements that we found with the work that we did was on the 62 hectares of trial sites that we had last year, we actually only saw a 1% improvement in use efficiency, which sounds really poor. What that doesn't show is the fact that we'd got some at 45% and some at over 120%. So the use efficiency was quite high where you got those exaggerated figures. What we've achieved this year is actually reducing that excess use efficiency. So we brought everything down. I think the maximum we got was about 103% use efficiency, 106 it says, sorry. And the minimum was 74. So we've had a significant impact. The average use efficiency across the whole group was 82%. So we'd actually increased it. While it's only by 1%, I think we've reduced the variability of the samples significantly and really improved things. It saved the farmers in the project across that area 5.4 tonnes of fertiliser, and it's certainly reduced the carbon emissions on the basis of that by 13 tonnes. That's all I'm going to talk about. At that point, I'll hand you over to Hugh, and Hugh will explain more about the carbon aspects and the measurements we've done there.
Thanks, Clive. Good afternoon, everyone. My name's Hugh Martineau. I am the Head of Sustainability at Map of Ag, and I've been undertaking some of the emissions analysis of the nitrogen use efficiency work that Clive's talked about so far. So greenhouse gas emissions impacts one element of what we've been doing, but I just wanted to sort of highlight the wider benefits of nitrogen use efficiency across the farming system. And sort of focusing around the pillars of sustainability, there are multiple environmental benefits of using nitrogen, and specifically reactive nitrogen, in a more efficient way. It also has an economic benefit, which Clive's already touched upon, and then it also reduces the wider societal impacts of nitrogen in the environment on things like air quality and water quality as well. Just to expand upon that a little bit further, so we have reactive nitrogen in multiple forms which leak from our biological processes and how we use it in farming systems. So we've got the nitrous oxide, which is a climate pollutant and has a global warming potential, but we also have nitrates, which are released into watercourses and have an impact on biodiversity there, and also ammonia, which is a contributing factor to biodiversity impacts and also human health impacts as well. So there's a much broader environmental impact of nitrogen in the environment beyond just the nitrous oxide impacts on greenhouse gas emissions. And I would argue that actually our management of nitrogen in farming systems is a sort of a clear route to sort of more sustainable and resilient systems. If we're able to work more within this system boundary as outlined here, so we're importing less nitrogen in the form of both inorganic fertilizers and feed into our agricultural systems, we stand a good chance of working within the system boundary more. I'll expand upon that a little bit more when I have a little bit look at some of the organic matter impacts as well. So just looking at the greenhouse gas emissions impacts on nitrogen use efficiency. So what we did, as Clive outlined, is a year 1 baselining study, followed by year 2 monitoring activities with some intervention, which Clive's talked through previously. So we've modelled the greenhouse gas emissions impacts on that variability in nitrogen use efficiency. But just firstly, I just wanted to highlight the relative share of nitrogen in its role in greenhouse gas emissions. So generally speaking, we see between 70 and 80% of greenhouse gas emissions associated with wheat and other cereal productions arise from the use of nitrogen. And a proportion of that is upstream of the farm in terms of the Haber-Bosch process of manufacturing nitrogen. And then a proportion of it is from the application of nitrogen in the farm system, which leads to nitrification, denitrification, and associated nitrous oxide losses. So moving into the results, and what do we see? How does this nitrogen use impact on the greenhouse gas emissions? So what we've got here is a situation where we've got emissions intensity up the side axis here. So this is emissions intensity as measured by kilograms of carbon dioxide equivalent per tonne of wheat produced. On the bottom axis, we've got the nitrogen use efficiency, which Clive's referred to. The green dots show what we had in the year one baselining study. So Clive mentioned we had 45% up to about 112% nitrogen use efficiency. So what that does in terms of the greenhouse gas emissions impact is significant. So we see the impact we see is around about between 150 kilograms of CO2 equivalent up to about 450 kilograms of CO2 equivalent between those two ranges. Now, what we succeeded in doing with the intervention trial in year two was actually narrow that boundary quite considerably. So we see that orange dots are where the activities of the intervention, so the leaf monitoring, and more strategic application of nitrogen over the course of the season and the impact that had. So the top of the area there, sort of, or the peak there, the highest emissions associated with these field trials was now 250 kilograms of nitrogen. We've got two dots outside the parameters of that 70 to 95%, which were actually an on-farm control that was done within that environment. So you can see that it's a significant impact on the overall nitrous oxide emissions from our production systems with the intervention trial in place. Just finally, I just want to touch upon the impact of organic matter and nitrogen use efficiency. I've been around a lot of tents here this morning, and I've heard carbon sequestration, organic matter a lot, but nobody I've heard so far has put it in the context of nitrogen use efficiency. I believe firmly that we should be looking at organic matter in soils as an agronomic benefit, not necessarily something to be traded outside agriculture. If we are in a situation where we are raising organic matters in soils, we have more ability and more resilience in the soil to deal with fluctuating prices of inorganic nitrogen, for example, because we should need less of it within the system. Now, if we build up organic matters over time, then we will see the opportunity to reduce overall nitrogen applications. And that is shown in some evidence done by a colleague of mine called Xavier Albano. He's joined us from Rothamsted, and this is his PhD study. I'm not going to go through it in lots of detail, but effectively this is a temporal resolution on his trial work, which goes from 2013 through to 2019, where he was applying different types of organic matter over the course of that period and seeing what the impact then was on nitrogen use efficiency. And the take-home message from that was, with the addition of organic matter, we were achieving higher yields with the same level of nitrogen. So there is a direct benefit of the additional organic matter in the system, so that we had a significant yield impact on that. So it's a really positive message, and I think that's really what we want to be focusing on in terms of the nitrogen use efficiency and the benefit of organic matters in soils. Just finally, just wanted to think about some future opportunities. We're in the year 3 analysis now. We've got some more innovation coming into the trial group now, people with companion cropping. We've seen significantly lower amounts of nitrogen used in this current growing season than we did last year, so it'll be really interesting to see the yield responses and the overall impacts on nitrogen use efficiency and greenhouse gas emissions as well. So that's the innovation point. I just want to sort of leave you with an idea that maybe through better data collection and precision, we might be able to detect over longer periods of time sort of in-field opportunities for different management. So whether different parts of the fields need more organic matter to be able to improve nitrogen use efficiency, or whether over time we've got areas of fields which are essentially just unprofitable and maybe more better suited to environmental management. So we get far more sort of mosaics of habitats and things within the field with that kind of diagnostic information when we understand in-field nitrogen use efficiency. So I'll leave you those thoughts and I think hand back to Duncan or directly to Alastair. Thanks.
Thank you both very much. Like I said, we're going to move straight on to Alastair now. I did introduce you earlier, Alastair, who's here. And then we'll have a Q&A panel at the end. So thank you.
Okay, thank you very much, Duncan. So I'm going to talk a little bit about building resilience through soil carbon, reduced tillage, and integrated pest management. Sometimes to go forward, we have to go back. This is the famous four-course Norfolk rotation. If you look at the graph, you'll see that you've got restorative phases and exploitative phases, and it goes round in a circle, and you match each one of those. And of course, we've forgotten to do that for quite a long time. We've just run endlessly exploitative phases. So we don't need to reinvent the wheel. Let's go back to the future. Here's some great work from Rothamsted. Where else? 180 years worth of stuff. If there's anything to be known about carbon, it's there. And you can see here a really good example of a grass field, which is the blue line, half of which has been ploughed out and put into arable. That's the hollow spots. That's the decline in organic matter when you go from a grass system to an arable system. The solid dots show what happened to the grass when it was continued to be grown as grass. Carbon continues to build up till you reach a certain point, and that's equilibrium. The red line shows an arable field, half of which is sown down to grass, and half of which is farmed arable. And you can see that when you put it down to grass, up goes the carbon again. And when you keep it as arable, well, it pretty much flatlines for carbon. So that's why the Norfolk rotation works pretty well. And you'll see that the carbon grows and declines very quickly, and then it levels off. So there's a lot to be had from short-term lays in terms of building back carbon and resilience into the soil. So this big project, you can see all the logos there along the bottom, lots of science coming out of it. That means we haven't got it now because it's coming from universities and they take years to write up their papers. But really good to see the depth of knowledge coming out of this, looking at multi-species lays, grass clover lays, looking at taking them, in fact, I can probably show you better on that one, taking them back into arable production sequentially and measuring the consequences of that. So, how long should you leave a lay in to get the maximum benefit? Because if you get 90% of the benefit in 3 years and only 10% more in 4 years, then you might as well just do it in 3. What happens if you plough it out? Do you lose a lot of the carbon? Yes, you do. Can you direct drill it? Yes, you can, but slugs and other things are going to be a problem. So it's how we work with that to take us forward. And that's what it can look like when you take nitrogen out of the system, and we didn't use any nitrogen in these trials at all. It's absolutely amazing what you can do in a low nitrogen situation, because clover obviously thrives and other legumes thrive in that low nitrogen situation. They have the advantage because they are fixing their own, whereas everything else around them has nothing. So they go very well. And just after a short period of time, if you look at the graph there, you'll see that the red bar is the all arable control, and the green and the yellow one is mint hill, and the other, one is the grass clover, one is the herbal lay. And you see on the left-hand side of the graph, we're measuring the carbon, but more importantly, perhaps, particularly given the weather today, we're looking at the permeability of the soil that carbon is delivering, and that permeability, of course, airspace becomes water space and then becomes airspace again, so the water holding capacity of that soil is improved as well. So that's less runoff during periods of heavy rainfall and more water storage ready for dry periods like we're experiencing now. So this is a bit of work we've been involved in. We're into our 5th year now, which is sponsored by Syngenta. We've got a number of sites around Europe. We're concentrated very much on 2 sites in the United Kingdom, one in Kent on a light soil and our own one at Loddington in Leicestershire. And we're looking at 3 tillage systems over a long period of time. So at Loddington, it's 3 tillage systems. You can see the 3 there on the screen. But we're going through a full rotation on each of the fields. So we're now in the 5th year of doing this, and we're starting to pick up some really interesting data, which is going to be helpful to people going forward. Here's some previous work prior to the study we're doing at the moment, just looking at earthworms. The increase in earthworms in non-inversion or non-plough tillage is, well, probably pretty well known now. On our own farm, the longest term field is 12 years into zero till now, and the earthworm numbers have gone from 200 to 700 per square metre. So you can just imagine chucking a bucket of water into each one of those holes, you know. It's going to go down far better in the one that has the earthworm tubes, that air space. And if you dig into the soil now, you can probably follow those earthworm holes down to 1 metre, even 1.5 metres in some cases, and you'll find the roots of the cereals using those earthworm channels to access moisture deep in the soil. And the last time we had a really hot spell in the summer, 2018, it was really noticeable that the fields that had more earthworms were the fields that stood the drought much better. And we're picking up some other things here. I won't. This is an aerial photograph of barley yellow dwarf virus, and you've got 5 replicates there. But I just ask you to focus on the ones in the top left-hand corner of the screen. You've got a plot which is very yellow with barley yellow dwarf virus next to one which has hardly got any at all. The only difference between these plots is the top one has been ploughed and the second one has been direct drilled. And we really struggled to understand what was going on here to begin with. I mean, we kind of convinced ourselves it might have something to do with beetles predating the aphids, which fly in and spread the barley yellow dwarf virus. But actually, it's got nothing to do with that. It's actually to do with spiders. And we found in one of the trials that we were doing there with Syngenta, where we have the light till, min till, and plough next to each other, we were growing oilseed rape. And we went in the autumn and did a suction sample, which is basically sucks the insects out of the crop. And then we analysed all those insects. And what we found was, so you have 3 bars there on the left, light till, on the middle, min till, on the right, you've got the ploughed one. And the yellow and blue section in the bar are the cabbage stem flea beetle species. So on the plough, you can see it's quite marked. In the min till, it's quite marked. But in the zero till, we couldn't find any cabbage stem flea beetle. So then we look at the orange section, which is the second one from the bottom, the bar above the blue bar. Those are the spiders. And you can see there are not many in the plough. There's a few more in the min till, but there's 10 times more in the zero till one. And what we think is that because in the zero till plot we're leaving the straw and we leave it quite long before oilseed rape to try and help keep the pigeons off, the spiders are spinning their webs and that is catching the cabbage stem flea beetle as it flies in. So there we are. We've solved the problem of growing oilseed rape without neonicotinoids. And so we repeated the exercise the following year, confident that we were going to be growing rape, and the spiders didn't turn up for work. And that is the problem with IPM. Unless we understand how to properly manipulate the systems to encourage this particular species that we want, it is going to be rather hit and miss. So we've got some more work to do there. And I'm pleased to say thanks very much to the funding from Kellogg's. We were able to draw on the substantial experience from the entomology department of the Game and Wildlife Conservation Trust, Professor John Holland, who has been working on this through his whole career, and we've put together this guide which provides guidance to farmers and growers as to what they can do to help enhance the number of beneficials on their farm to help natural pest control. I think I've probably finished that point. Sorry? Well, I guess I could say a few words about hedgerows. Yes. Right, okay, there we are. Let me just skip through those because, okay. I'll say a few words about hedgerows. Yeah, we're also working at the moment on a hedgerow carbon code. This will allow you to use the land app to measure the length of the hedges on your farm, put in the width and the dimension, and it will give you a carbon figure for that hedgerow. You can then project how you're going to allow that hedge to expand, or in the case of a brand new hedge, plant a new one, or in a case of a hedge which is gappy, gap the spaces up. And that will allow you to calculate a carbon figure. And again, this is another project we're working with Kellogg's and with the farmers that are part of our Origins group. We've now created the software that allows us to do the calculations, and we're just starting to pilot it on some of the supply chain farmers that are in the group to see what sort of carbon numbers we've got. It's a very interesting study because we're also looking at some of the carbon in the soil under the hedge, which is on occasions more than is actually in the hedgerow. So ultimately, we'd like to create a hedgerow carbon code which allows you to calculate all the carbon in the hedge space. And hopefully then we can trade our grain with Kellogg's and the carbon credits with it, and that will offset the natural gas used to cook the Special K in the ovens in Wrexham, keeping it all inset within the system. Have I done it?
Okay, thank you very much. And the nature-based solutions guide that Alastair referred to, this one, I think we've got a few copies that are available to hand out over there somewhere. So please grab one of those as well if you want one, along with the nitrogen use efficiency work as well. We've got a good slug of time now for Q&A. So please, we've got a roving mic just over there. So hands up if anybody's got a question. What's it? One in the front row, please.
Thank you for those wise words, and I look forward to carbon neutral cereal from your Special K. Question on the nitrogen, was that all solid nitrogen?
No, solid and liquid.
And liquid? Yeah, and differential between the two?
Didn't see any.
None?
No.
Thank you very much.
So no, there was no difference between solid or liquid applications. We had farmers using both, and there was literally you couldn't see any difference. I think there might have been some small difference in nitrogen uptake on the liquid in the drier conditions, but it was marginal. It's hard to. I couldn't say I've got evidence to prove that.
Okay, Dave.
Thanks, Alastair. Just unfair question on the start of your hedgerow project, I think. But just wondering, on the carbon code, are you considering additionality? So obviously you have a hedgerow there, it's been there for a long time, the carbon's been there for a long time. How are you going to start to trade that carbon if it's essentially net no new carbon if we're still managing a hedgerow?
Well, I deliberately didn't use the word trade, actually. But that actually won't be down to us. So within the project, we have organic farmers and growers who are the auditors for the Farm Woodland Code and the Peatland Carbon Code. You're quite correct. At the moment, everything should be additional. And when we started on this project, we had actually a particular group of hedges in mind to target. These are hedges that are on now all arable farms, which were formerly mixed farms. You can't pull them out because they're protected by the hedgerow regulations. So they're usually managed to within an inch of their life through flailing. And that's because they are of no value to the farmer. In fact, they are an inconvenience. And yet they can deliver a great deal, not only in terms of carbon, but biodiversity as well. And we were interested first and foremost in the biodiversity. But it struck us that if you could add a carbon value to that, and trade some biodiversity on it, then all of a sudden, farmers will start to look at that as an income stream and worth actually expanding. So that's really what we're aiming at. And well, you've visited Loddington, it's quite lamentable that our own farm is not going to be able to benefit massively out of this, because so many of our hedges are already grown out for biodiversity. There is some discussion within the Woodland Code at the moment about how existing woods might be improved to up the amount of carbon locked up in them rather than leaving them unmanaged. And I suggest there might be some space there for a discussion about hedgerows, yeah.
Thank you. We've got a question right at the back, and then we'll come to you, sir.
Thank you all. Regarding nitrogen use efficiency, any indications whether that is a genuine improvement in the use of the nitrogen you apply out of a bag, or is it using the nitrogen that is residual in the soil? If the latter, could you ultimately deplete the nitrogen in the soil?
So one of the aspects that we've introduced to the trials this time and work that I've been doing separately on another site, we've been historically measuring the soils. So we understand what was in the soil. Certainly after harvest, we are seeing a lot less nitrogen left in the soil. So we are taking some of that out. It's pretty early days yet to have a significant data set to show whether it's actually coming from the soil or it's just residual that was left from fertiliser in previous years. And part of the work has been looking then at organic matter and carbon sampling within that layer, so that we can qualify whether or not we're degrading the organic matter levels that could be influencing nitrogen losses. So it's too early to say. We've got questions about it. It's something we don't understand. The suspicion is that we are utilising more effectively the fertiliser we're applying, and we certainly are having soils with less residual nitrogen after harvest. And to be clear about the protocol, we're sampling before harvest. We're sampling in autumn and again straight after harvest. So there's no mineralisation that could occur after that once the crop's removed.
Okay, question just here.
Clive, on that point, the variability in sampling. So we have sampled our soils and we have got loads of maps showing indices of all kind of nutrients. We also started to look at the carbon, but actually what you're saying, a carbon map or soil organic stroke carbon map is much more accurate to apply your nitrogen and nitrogen release. The variability in doing it, because one of the things we've never really found the exact measure, and it relates to the time of the year as well, doesn't it, to do it. So have we found the science, what to test the soil for in part of the rotational system that's in, its ability to release nitrogen at the right time, because that's one of the other issues, isn't it, that we come up against. So are we any closer for having a protocol or a template for what we should do?
No, I think that's still very much a minefield. So the sampling regime, I agree, you've got a lot of variability across fields. So I don't use, the methodology we're using in the trials is very, very, very small areas. So literally the patch of chairs that you've got there that you're all sat in would be bigger than one of our trial areas. So we're taking the soil samples from a very small area. We're doing the leaf samples above that area, so we know exactly what's in there, and we've got very limited soil variability there. It would be sampled on a conventional basis where we would take at least 16 cores. We were sampling down to 30 and 60 centimetres, and we were also doing nutrient analysis within a broad spectrum nutrient analysis in the top as well. The sampling depth is critical, and the quality of sampling is critical. So what I initially found was 3 years ago on the farm at home, we've been sampling quite regularly, looking at organic matter when we're into no-till, and we've steadily seen in a conventional soil sample in the surface, organic matter levels in the soil, in the topsoil, rising. And being very proud of the fact that we're rising it. I was horrified when I did a carbon and organic matter sample down to 30 centimetres, that on all those sites I thought I was getting an increase in soil carbon and soil organic matter. They actually dropped back to the levels there were before we actually started doing min-till. And I'm pretty convinced that process is migrating organic matter into the surface because we're not incorporating it. So the sampling process is really critical to make sure you're doing things consistently. A small change in depth of sampling for nitrogen or organic matter, if you do it conventionally, could potentially have a huge impact on different cultivation and establishment techniques to the results that you get. So everything we're doing at the minute is 0 to 30, 30 to 60. It's in a small area, and we go back to an area and we've tried to remove the whole variability question of the soil out of it. The challenge we've got is understanding where's the best place to put that sample point and make it representative of the field. And I haven't got an answer for that properly yet.
Okay, question here.
Yeah, thank you. I couldn't quite see the colours on the graphs about the organic amendments and the nitrogen use efficiency. Does the C/N ratio of the amendment play a role?
Very much so, yeah. It's something I should have pointed out on the slide, sorry. So one of the organic amendments was incorporated straw, which you saw actually nitrogen use efficiency below the control because of the carbon-nitrogen ratio. But whereas we've got a more appropriate carbon-nitrogen ratio with farmyard manure, anaerobic digestate, and compost, then the benefits were seen in those environments where you've got an appropriate carbon-nitrogen ratio. But yeah, it's a very good point to point out. Thank you.
Okay, thank you. Phil.
Yeah, question for Hugh again, based on that. I take it some of these organic amendments also probably had some nitrogen and other nutrients in them, so there would have been multiple benefits of why it was increased. And I have got a second follow-up for the esteemed panel, and that is, are we likely to see listings with nitrogen use efficiencies on wheat and barley varieties? Will it make much a difference?
So I'll start on that. The benefits of the organic matter in the trial showed that if you're applying the same amount of nitrogen, you were getting a yield benefit. So you had a yield benefit over and above the nitrogen application with the organic matter. That was the key take-home from that. But it acquired over time, so you've got the build-up of organic matter over years. So you didn't get the benefit in year one, but in subsequent years, you're starting to see an increased benefit of that. So the take-home was really about the additive effect of the organic matter over and above. So if you're applying the same amount of inorganic nitrogen without it, you didn't get the same yield. You saw a yield penalty in that environment. Is that clear?
I'll take the second point there, Phil, because I think you've been monitoring my inbox. It's an interesting comment, and I asked the same question of the HDB last week because the new recommended lists are out for this year. Currently, you can see grain yield in there or variance from normal. You can see grain protein there. I asked the question, what's the nitrogen applied to the different trial sites, and was that available? So we could try and I wanted to try and do a variety use efficiency measurement to see if there was a big varietal difference. And while there are protocols, each trial site has its own protocol for nitrogen. It's done at farm practice based on the RB209, so they're all different. It is in the protocol to record the nitrogen that's applied, but it's rarely been asked for, and there's very little data actually recorded at HDB on the nitrogen that's been applied in detail, when, rate, product, how much, in a lot of detail. And it's going to be quite difficult to do that initially with the historic data sets. But it is something I'm certainly looking at, and I think you're right. I think it should be a factor that's presented for growers to make a decision on.
And I'll take the bit for the esteemed panel, if that's okay. We did some work with Harper Adams for a bread maker, and we looked at the scavenging ability of different varieties to be able to access the nitrogen pool in the soil through the root structures. And there's obviously no question that the better root structure that you've got, the better you can scavenge the nitrogen. And I think this is one of the problems of all the trials being done at optimum nitrogen levels, is it doesn't make the plants seek out the nitrogen. I know a lesson I learned when I farmed organically, that if you've got any compaction, my word, it doesn't half show up quickly when you're not putting back nitrogen on. Yeah.
Okay, thank you. A question at the back.
Now, in order to get away from the Haber-Bosch process, firstly, is any work being done on finding ways of getting nitrogen-fixing nodules onto plants other than legumes? And secondly, what happens if you plant wheat into an established low height of white clover?
I can't answer the genetic bit. I'm sure there's some work going on somewhere, but I'm not qualified to comment on that or know where it is. On the second point, one of the growers in the Kellogg's Origin group has actually got a trial site this year with undersown clover. And we've actually been monitoring that site as well. So that's something we're learning. What we learnt initially was the wheat was very deficient. We think the clover was actually probably being detrimental to taking nitrogen up initially. And it takes a while for clover to establish and the nodules to get going. That said, the readings that were taken with the end tester recently, the clover field is has got the highest leaf end concentrations according to the end tester at the minute. So there's definitely something there. We've got a lot, I think, to learn with that process and how we establish crops and maintain that clover sward in the bottom. But quite how we manage that properly, I think we've still got a lot to learn.
I think given that 40% of global calories come from the Haber-Bosch process, it's going to be a very long time before we replace that fully. I think perhaps more exciting is the prospect of using green electricity to hydrolyze water, split the hydrogen atom off, and use that in the Haber-Bosch process instead of splitting the hydrogen off methane, natural gas, which then leaves you with the big CO2 footprint. That, to me, looks encouraging. Unfortunately, you need ammonium nitrate prices to be about £800 a tonne before that becomes applicable. But hey, you know, that's not beyond the realms of possibility, I believe.
Okay, question over here.
It's not a question, it's just a quick point to have a question about putting rhizobium onto other plants. There's a lot of work going on that, but it's looking like then when you introduce nitrogen fixation into species that don't normally do it, there's a massive energy cost to the plant. So you get an associated yield loss. So I think my understanding, it's not my field of expertise, but my understanding is they're looking at more marginal land like Africa, etc., where it may be beneficial. But I think in the UK environment, we're a very long way away from that.
Okay, any other questions?
Just going back to your nitrogen use efficiency, have you looked at urea and some of these inhibitors that are about for urea? It's quite topical at the moment with them trying to ban urea, and just wondered if you got any comments on that.
I don't think we've got any growers currently that have used urea. Well, not in a direct form. Some of the liquid guys will have used UAN. But it's not something I've got a lot of evidence on, and I certainly have no evidence at all for the pros or cons of inhibitors in there. I've got personal opinions on that, but I'll not share those.
Question down here.
Can I just pick up a little bit on that? Just in terms of the role of inhibitors, I think they have got a role to play. Until they sort of become cost effective, I think we're not necessarily going to see them widespread in use, but they've certainly got a role in reducing the amount of nitrous oxide we see released from nitrogen applications.
Yeah. Have you looked at the work by Lauren Steinledge in the US, who's interseeding legumes and wheat together, and he's getting the same yields from the crops by putting these two crops together and then taking them off with different heads?
Not recently. I have seen it. I saw some of that work done in 2004 around Lincoln in Nebraska. They were doing that sort of thing. I've not followed it recently, but they were growing crops of wheat there as a break crop to try and reduce nitrates in water, and they weren't applying any nitrogen to the wheat at all, and they were achieving 4 tonne an acre equivalent wheat crops with no nitrogen. But I think that was just a sorry state of the status of the water courses and the amount of water that was polluted with nitrates.
You know, I'm just surprised I haven't seen more in the UK the inner seeding of these crops together to get the nitrogen from the legumes.
Yeah, I think there's one or two starting to look at the polycropping concept. I think one of the enablers that is available to farmers in North America that isn't available here is the genetic modification. And the fact that you can then do weed control, for example, with Roundup, so that you've got the ability then to control weeds in a better way. But it's, you know, that was one of the enablers that let that technology or that methodology work. I think we've got a lot to learn on how we can do that conventionally.
I don't think it's a new concept. I mean, it's just something undersowing spring crops. We do it at home, spring oats with white clover sward, and we've used very, very little additional nitrogen in that system. It's not a new concept, and I think it's got an important role to play.
Okay, Rebecca.
Thank you. Question for Alastair. How do you anticipate the new hedgerow carbon code translating into a future SFI standard?
We haven't been asked to do that, but you make a really interesting point. Because, of course, you know, if we're going to start trading biodiversity benefits and carbon benefits from hedgerows, then the Treasury's going to start to get interested. You know, the whole purpose of the stewardship scheme is to pay public money for goods that the commercial sector otherwise wouldn't perhaps fund, and to fill that gap or to pump prime it. But, you know, the Treasury are pretty sharp, and if they see farmers stacking public money and private money and making vast amounts out of it, then they're going to come back looking at it. Now, let's be perfectly clear, we are a very long way from doing this, and we've been telling government that they don't want to even be thinking about this at this point. If farmers get a sniff of the word clawback, then the enthusiasm for entering to the scheme evaporates, and government have an ambition that 70% of people go into the new environmental land management scheme. So let's not shoot ourselves in the foot from the start over something which actually may never come to pass.
Okay, any further questions? I've got one, if I may, Alastair, which is quite a broad question. We've done some great work, or you guys have done some great work on the habitat management piece, which is, you know, identifying what sort of habitats attract the right sort of insects that you want at the right time. Can we ever realistically get to a point where we don't need to use insecticides at all, that we can actually manage the insects in such a way that we reduce our reliance on insecticides?
I would never wish to say never on this, and you know you can get a plague despite everything else that you've done, and then I think it's always useful to have the final armoury in the toolkit, which allows you to take a targeted intervention. So I don't ever see that in the short term, at least, being the case. But there are a lot of ways that we can be smarter in what we do at the moment. We take a very broad acre approach to cropping, and I think we can be smarter.
Mm-hmm. Okay, any final questions for anybody? Okay, I'll draw this session to a close then. I just want to say thank you very much to our three esteemed speakers. Hugh, Alastair and Clive, thank you very much for imparting the knowledge today. And I'll just say thank you again for attending this session, and just how delighted we are on behalf of Kellogg's that we are here, been able to sponsor this tent today and be part of the whole Groundswell movement, I guess. So thank you very much. Thank you.